So, guys, we are going to discuss today about the body fluids and circulation. Let us see during this revision session what are the extra things that we come to know. So, first we have to understand what is circulation. What is circulation? Circulation means transport of substances throughout the parts of body of organism. So we know that circulation is required for unicellular organisms like small amoeba or paramecium. You must be wondering what you are saying, ma'am. If they are unicellular, why they require circulation? They have only one cell. But beta, for them that cell itself is quite a big. So for unicellular organism, the intracellular circulation is there, which is done mainly by a method called cyclosis, or it is also known as cytoplasmic streaming. So what is this? Actually, cytoplasm present in their cell shows regular streaming movement. What do we mean by streaming movement? That means that cytoplasm will turn around at different corner of cell in a cyclic pathway. So from this point, if it is starting, consider this is the cell. If it is starting from this point, it will move like this and come back here. So this is an excellent way of communication. Apart from that, we all know that ER and lysosomes, they are also responsible for transportation along with Golgi bodies. But when we are talking about extracellular transportation, it's not that easy. Why? As organisms grow big, that means in their case the matter is more complex. We already observed that as organisms go big, the cells present inside are never coming in contact with the surrounding. They are inside. So if oxygen is absorbed from somewhere or food is reaching to a specific part, it has to reach to every part of the cell. And not only positive things, it's not only the positive things which is done by this circulatory system, the negative things also should be removed, like excretory substance. Apart from that, communication made in our body via hormones and via other chemicals, which are also transported via blood circulation. So we have to know that what are the extracellular circulation. Among that, the first one is water circulation. That means the environmental fluid circulation. So can you give me example of any animal where water circulation is there? Guys?
Sponges.
So sponge, hydra. So as they are very small and almost all body cells are in connection, some from the surface, some from the canal system. In sponge, many canals are there, branches, and it's a well-developed structure. In case of hydra, single canal is there. So water goes in and out, and the body is bathing in the environmental fluid. So that's why water circulation is also a kind of environmental fluid circulation. Now, second is body fluid circulation. So in this case, no external thing, the fluid present in our body is responsible for circulation. So what is happening in this case? In this case, we will first talk about the parenchymal circulation. So do not get confused. Parenchyma is present in case of animals also. So it is found in case of flatworm like liver fluke. So what they do, they actually do not have any kind of body cavity. So the space between body wall and the internal organ is occupied by a tissue which is called parenchyma tissue, which is having large irregular cells, and the branching is there, and the whole body is filled of fluid filled spaces in between. So whenever the digested food is there, it is diffuses of the parenchymal into the parenchymal fluid, and contraction of the body cause a very irregular and slow movement of the fluid, so that food material can be distributed to the body cell. So in this case, the body fluid is composed of parenchyma cells. That's why it is called parenchymal circulation. Obviously, you can understand it is very inefficient one, and as these platyhelminthes or flatworms are very small, so they manage with it. So next is body cavity circulation. So in roundworm, they have body cavity. What do we call animals with body cavity? So they have body cavity, but this is not the true
Coelomate.
Nana. They have pseudocoelomate. So as they are pseudocoelomate, the body fluid is known as pseudocoelomic fluid. Understood? So they have false coelom or pseudocoel, and it is having pseudocoelomic fluid which is taking responsibility of the circulation. And in visceral part, they have like very minimal kind of muscle like structure through which the body wall contract and expand, and that cause a minimum movement of the pseudocoelomic fluid. Okay. Third one is the blood vascular system. So we have the blood vascular system, which is the most developed one, and we have heart. But remember that higher invertebrate onwards we have heart, but all blood vascular systems are not that well developed, because some may be having open circulatory system which are not that efficient. Apart from blood vascular system, we have the second vascular system that is known as lymph vascular system or lymphatic system. Understood? It is having white fluid which is called lymph, and its main function is to give us the protection against the diseases. All vertebrates are having this lymph vascular system. Understood?
Yes.
Yes, ma'am.
So our whole circulatory system is developed from mesoderm. As we are coelomate, we have well developed ectoderm, endoderm, and mesoderm, and that's why we are our whole circulatory system. This together we call as circulatory system. is derived from mesoderm. Okay. Now we will talk about the blood and lymph system. So, what are the components of blood vascular system? Anyone? Blood vascular system is made up of what?
Yes, ma'am.
I am asking for what? Yes.
Arteries, veins, capillaries.
Very good. Arteries, veins, capillaries. So first,
Lymph
blood vascular
vessels.
blood. Isn't it? We are having closed circulatory system. So what is the difference between open and closed circulatory system? Let me first talk about open circulatory system, because in case of open circulatory system, there is though no proper demarcation between the tissue fluid and the blood. Why? Because blood vessels are not intact. That's why always the blood is coming in direct contact with the tissue. So it is pouring the blood into sinuses, and blood is coming under direct contact with the tissue fluid. Is it good or bad? Open circulatory system is good or bad?
Ma'am, it is a bit primitive, so it might be harmful.
हार्मफुल तो नहीं है, जिनके पास होता है दे आर हैप्पीली लिविंग विद इट. बट माय क्वेश्चन इज, व्हाट डू यू थिंक, इट इज गुड और बैड? Like, is it more useful than the closed system or less useful?
Less.
Useful.
Why?
Energy supply
Ma'am,
is
because
less.
there is no specific demarcation for oxygenated and deoxygenated.
That is called mixed blood. Do not confuse both the things. We are talking about open circulatory system. Even in closed circulatory system, it is possible that oxygenated and deoxygenated bloods are mixed. That occur in case of fish and amphibian, except crocodile. So one by one, what is the disadvantage or advantage of open circulatory system?
Ma'am, everything is bathed in the cavity, so it might contain everything, dirt and the useful products too. So
So what is problem associated to that? It can, no problem.
Ma'am, energy supplied is less.
Very good thing. That energy supplied, we should not say. The speed of transportation is reduced. That is what is the most important thing. The action, because our circulatory system is enclosed in a pipeline structure, that is blood vessels. So obviously it is speeding up. But if consider in blood vessels, in between there is gap where blood is directly coming under contact with the tissue, obviously the speed and accuracy will be less. But the advantage is they don't require specialized protein for transportation of O2. So generally, even if they are having specialized protein like hemoglobin is there, hemoglobin doesn't require specific cell inside which they can be there. Hemoglobin generally can be present easily in the blood plasma, and then it can be bathing, the whole body can be bathing, and as whole body can be bathing, the hemoglobin is able to supply oxygen from plasma itself. Okay, so we often use a word called hemolymph. So what do we mean by this hemolymph? Hemolymph means in case of organisms with open circulatory system, they don't have clear demarcation between the what I can say, the blood and tissue fluid. So altogether, all of these are called as hemolymph. Okay, and the cavity through which it flows is called hemocoel. Okay, the tissue space or the space between the tissues through which it flows is called hemocoel. So kindly note it down, you may get confused. Next is closed circulatory system. So we are going to talk about it. That's why if anyone asks you about the components of blood vascular system, you should first talk about blood. Okay. Then you should talk about the heart. And third thing you should talk about is blood vessel. We discussed about composition of blood in detail in tissue chapter. You want me to repeat it once again?
Yes.
So as I suggested that in case of closed circulatory system, every organism will have better efficiency of circulation in terms of speed, reachability, and we can regulate the blood flow. You must have seen that whenever we are running, our blood gush, but whenever we are come down, the blood flow is not there. So that means unnecessary, we don't allow our blood to run around. Depending on situation, we allow it to move here and there. So I will start with the like blood. As you said, in case of blood, if I talk about the blood, study of blood is known as hemology, because hem word is related to heima, H-A-I-M-A, which is in Greek means blood. So if you look into the blood, it is an opaque fluid and it's a fluid connective tissue. As I said, the components of this circulatory system is made up of mesoderm. Blood is also no exception. And it is like having slightly heavier specific gravity than water. For water, it is always 1. In case of blood, it is 1.06. If you taste blood, I guess everyone must have tasted their blood once or the other. कभी भी हमारा उंगलियों में कुछ चुभ जाए, we always have a habit of sucking the blood. So you know that it is salty in the taste and it is mildly alkaline because its pH is 7.4. Okay. So what is the color of blood?
Red.
If it is a fresh red, that means it is well oxygenated, it is bright red. And if it is deoxygenated, it will be having a purplish color. Okay. So blood can be further subdivided into 2 components. One is the liquid part, which is known as plasma, and other are the cells of blood. So these cells or the blood cells are floated in the, they are present in the plasma, they are carried by the plasma. So as I said that the all connective tissue is having a matrix and cells are present inside this. So blood is a liquid connective tissue. So plasma is the matrix. So many a times in exam, one very good question is asked about the difference between serum and the plasma. So we will discuss about it later. It's a very important question. You can mark it down about the difference between serum and plasma. So what is plasma? It is a slightly alkaline yellowish viscous fluid, and it is always in an equilibrium with the tissue fluid. That's why if there is any kind of abnormality in our body, its trace is first time found in blood. इसीलिए doctors, whenever they have a doubt, they go for testing of the blood. If a person is having hormonal imbalance, it can be detected by blood. Any kind of foreign substances will secrete certain kind of chemicals. Its presence can be detected by the blood. If there is any kind of metabolism disorder, that can be also detected by the plasma. So blood plasma is having 90% of water. Okay, and 1% of it is inorganic salt. Anyone guess what it is? Apart from that, 7 to 8% of proteins. So, and that is the beauty of blood, that for inorganic salt, it is solubilizing it as a normal solution. But when I am talking about protein, they are present in the blood as colloidal solution. I guess everyone knows the difference between colloidal solution and solution. You must have studied it in grade 9. Okay, so is 100% done or anything is left? Class?
Ma'am, formed elements.
No. Is 100% over 90 plus 1 plus 7 if you consider? Some amount is left, yes or no? Out.
Yes.
Yes,
So that 1 and 2% is made up of food, on it can be excretory substance. Apart from that, it can be dissolved gases are there. What else? What do you think it can be there? There can be regulatory substance.
Hormones.
That is, I am making it all together about all substance that is regulatory. Apart from that, any guess, anyone? There can be antibodies. Although they can be counted as proteins also, and there can be cholesterol. Okay, there
Nutrients.
obviously anticoagulant. So what is the anticoagulant we have in our
heparin.
Heparin.
So that is also present. So this is the rough composition of the plasma. So now I wish to speak in detail about what are the proteins present in plasma. In plasma, there will be presence of serum albumin, and there can be serum globulin. Out of this globulin, immunoglobulin is one. Then there may be properdin, prothrombin, fibrinogen. So all these are acting as a protein. So plasma is basically not only supporting the blood cells and giving them a place to live, it has many functions. So basically, it's a source of protein for so many types of tissue cells. And these source of proteins are many times precursor, and they provide the tissue cells to synthesize their own protein from these precursor. Apart from that, plasma is a very good buffer. The buffering activity of blood is provided by plasma because cells cannot give them the buffering activity. Apart from that, albumin and globulin present in the blood maintaining the osmolarity of body. And if there is fall in the plasma protein, that will be causing the problem. And if there is falling of the plasma protein, we may have edema of hands and legs. So what is this edema? It is although spelled as OEDEMA, oedema, but the pronunciation is edema. Edema means swelling. Okay. Apart from that, you know that plasma protein may be transport protein. They help us to fight against diseases like antibodies, and protein present in the blood can directly destroy some bacteria and neutralize some viruses. Apart from that, the inorganic salts present in the plasma are maintaining the ionic balance in the plasma. For example, sodium and chlorine ions present in the plasma is very, very important to maintain the ionic balance. But apart from that, bicarbonate, phosphate, potassium, magnesium, calcium, iron, manganese, they are also present in very small amount and they are also crucial. These are altogether called blood electrolytes. And see, Rutu was talking so many times about the formation of urine. So their balance should be maintained in the blood. If NaCl is reabsorbed, it should not be too much reabsorption, it should not be too less reabsorption. We know that blood is having good source of food material. So why? Because after digestion, the food is digested, food is carried to the different part of our body via blood only. Same is for waste products. It has to be displaced from the cells unless there will be uremia, and dissolved gases will be there. Regulatory things like hormones, enzymes, and vitamins should be there. Anticoagulant, which is called heparin, is there. You talked about heparin, but there is other way of stopping anticoagulation, stopping coagulation. Can anyone give me name of any other anticoagulating substance? Anyone, any idea? Heparin is having other name. Heparin can be called with same chemical, but it has another name to be called. It is also called antiprothrombin. Understood? So it has cholesterol also. So cholesterol is good or bad?
In proper amount, it is good.
So actually 50 to 180 milligram per 100 ml of blood is good. If it is increasing further, it is not good. Okay. Antibodies are there. So that's all I wish to talk about the plasma. Now I'm going to focus on the blood cells. So this part is also taught to you quite in detail. So I just wish your response more in terms of blood cells. So
Ma'am,
yes, beta.
so our body have only one anticoagulant, that is heparin or antiprothrombin.
Yes. Too many of them will be confusing, but it has two names. That's why I mentioned. Okay. So it has RBC, which is full form is red blood cell. Its good name is red blood corpuscle also we say, and its good name is
Erythrocytes.
okay. So this is one of the most abundant blood cell present in our body, and they are like what I can say, most important cells because they are responsible to carry oxygen. So if you look into the shape of RBC, they are generally like oval and biconvex. In specifically mammalian RBC is biconvex, means if you look into the structure, it will be having structure like this. The margins will be slightly elevated and in between there will be one depression. But in case of amphibians, reptiles and others, you can find variation in the shape of RBC also. They can be oval, they can be nucleated also in some cases. But in case of mammal, the mammalian RBC is always biconcave and it is non-nucleated, because the thing is that for increase the efficiency of transportation, one needs to have more space, and once the cell is produced and it doesn't have to divide once again, because new RBCs are not produced from the old RBCs, they has to be produced from the stem cell present in the bone marrow. Then for what purpose they require nucleus? They has to, once the hemoglobin is synthesized, the RBC is matured, they need to only go for transportation. So to maximize the what I can say, efficiency of circulation, their nucleus is gone. Okay, and this biconcave shape of RBC present in human is a shape which is actually very efficient one. Why? You know, when RBCs move in the blood vessel, they can line up like this, and then they can move one by one. Apart from that, this shape is helpful for, not only helpful for easy movement, it increases the maximum surface area, because if we talk about the sphere, the central part of sphere never comes in contact with the diffused gases. And if I'm talking about same way biconcave shape, it is having more and more surface area. So their size is approximately 7 to 8 micrometer, and the lifespan of RBC is approximately how many days?
120.
120 days. Okay. So if I'm talking about the number of RBC, the number of RBC is 5 to 4.5 cubic millimeter per or rather to say, let me write it properly. It will be 4.5 to 5 million per cubic millimeter. That is, I can write it like this also, blood. So you can understand their number, how it is the maximum. Out of the all blood cells, the RBC number is highest. So if there is decrease in the number of RBC, we call that disease as, can anyone tell me? Very common disease.
Yeah.
What it is, beta?
Ma'am, would you please repeat the question?
I asked that if there is decrease in the RBC number, what that disease is called?
Ma'am,
Hemophilia.
Hemophilia doesn't have anything to do to RBC, beta. It's called anemia.
erythromia.
Anemia.
Okay.
Simple name, beta. Okay. So, Why it occurs? What do you think that anemia occurs? It can be by
Ma'am, when there is excessive blood loss,
yes.
if someone meets with an accident.
Apart from that, there can be hemolysis, that due to some certain reason the RBCs are broken down and lysis. There may be a disease where blood cell formation is faulty and the person cannot go for it. Apart from that, if a person is having vitamin B12 deficiency or iron deficiency in the body, then also a person can have anemia. Okay, so in our body, do you think RBC count ever increase? Do we have ever the increased RBC count in natural condition? Anyone?
Ma'am, in the embryonic or fetus phase.
That is there. Very good, Rutu. But in normal life also, you must have seen certain people are fit. And why they are fit? When you exercise regularly, the RBC number in a person's body increase. Another example I wish to quote over here. You must have seen that in hilly region, I taught this to you earlier also, that in hilly region, person can very comfortably climb the mountains and like without, they don't require any specific gear because it is their natural tendency that they have more RBC in their body. That's why with the thin air or the less oxygen volume also they can breathe very easily. But there is a bad thing. Sometimes there may be abnormal rise in RBC, and this is called polycythemia. I'll write the name of disease. It is called polycythemia. Okay, so in polycythemia, um, um, there will be increase, and there is another disease where there is abnormal lowering in the RBC. That is also called erythrocytopenia.
Erythropenia.
Erythrocytopenia. Okay. So we have in our body the like mechanism to combat this erythrocytopenia. How? The moment there is lowering in the blood oxygen level, uh, the kidney will secrete the hormone which is known as erythropoietin. And erythropoietin hormone will stimulate the bone marrow so that the RBC production can be increased. Okay. So once the RBCs are increased in the blood and that they are reached to the adequate amount, the blood oxygen level will become normal and the erythropoietin synthesis can be stopped. Okay, understood? Guys?
Yes, ma'am.
Great.
Yes.
So RBC actually looks yellowish, and if we look into them in a bulk, we see them red. Another reason is the oxyhemoglobin formed is red. That's why RBC looks red. So basically, the red color of blood is due to the presence of hemoglobin. So it's a conjugated protein, that means quaternary structure, and conjugated because it is having iron at the center. So it has a heme unit which is having iron, and it's a basic protein which is called globin, and the non-protein unit is heme, which is the inorganic unit. That's why we call it hemoglobin. Okay. The heme is the iron porphyrin ring. So where we studied about porphyrin, Apart from here, have you-
Chlorophyll.
very good, very good. So, 4 globin molecules are there, 2 alpha, 2 beta, and they are attached with the porphyrin ring or heme to form the hemoglobin. So, there should be 12 to 16 gram hemoglobin present in per 100 ml of blood, and this is the adequate number. So, if a person is having less like amount of hemoglobin, that is also detrimental. In spite of having RBC, if the amount of hemoglobin present in them is less, that is also problematic. We already talked a lot about the formation of carboamino hemoglobin and oxyhemoglobin, so I am not talking about it over here. So, what is the process of blood cell production? Can you give me the name of process of, or rather to say process of production of RBC? It is called erythropoiesis. And in our body, the B12 and folic acid stimulate the erythropoiesis. So you said yourself that blood vessels are responsible to carry hemoglobin for 120 days. After that, they die. So what happens once they are died?
Ma'am, they go to their graveyard, that is spleen.
So that's why it is known as graveyard of RBC. So after going there, what will be happening?
Ma'am, the
मैम वो
blood cell
वहां
will
से
break
लीवर में
down in
One by one, Ayushi said so many, now Rutu can speak.
मैम मतलब वो वहां से उनका कुछ प्रोसेस कंप्लीट करके कुछ पार्ट्स जैसे हीम है हीमोग्लोबिन वो बिलीरुबिन और बिलीवर्डिन में कन्वर्ट हो जाके लीवर में चला जाएगा.
So that is true, and the protein part can be also reused. Okay, so that is very important thing that is present in this RBCs. So now we will talk about the next type of blood cell, that is WBC, white blood corpuscle, or we call them leukocyte. Do not get confused with the lymphocyte and leukocyte. The lymphocyte is a type of leukocyte. So they can be of various shapes because WBC can be of various types. So their number varies from 6,000 to 8,000 per cubic millimeter of blood, and it depends on like how their what type of WBC they are. So similarly the way erythropoiesis is there, leukocytes are produced by leukopoiesis method by from the same cell, that is the stem cells of the bone marrow. So if there is increase in WBC count, we call this as leukocytosis, and if there is decrease in blood with WBC count, we call it leukopenia. Understood? So leukopenia is decrease and leukocytosis is increase. Is it clear?
Ma'am, would you please repeat?
Leukocytosis is the rise in WBC count. Okay, and it is generally occurring whenever we are having some kind of disease or infection or inflammation that is signed by itis. I told you na, pharyngitis, rhiningitis, meningitis, any itis or inflammation, the number of our WBC increase in our body, and that process is known as leukocytosis. And the way in erythropenia there is decrease in RBC, in leukopenia there will be decrease in the WBC count. So whenever we are infected with AIDS, which is causing havoc for what I can say, um, destruction of immune system. At that time, we may have leukopenia. WBCs are actually not white in color; they are colorless, and if we keep it in contrast with the RBC, actually that's why we call them white blood cell. But they are not. मतलब white color means no color. Understood? Is it clear?
मैम
Yes, beta.
मैम जैसे किसी को अगर ब्लड में इंफेक्शन होता है तो कई केसेस में बोन मैरो चेंज करता है, तो जैसे अगर वाइट ब्लड सेल्स को कोई चीज इफेक्ट कर रही है तो उस फैक्टर को हम नहीं निकाल सकते?
नहीं, ब्लड में इन्फेक्शन होने की वजह से बोन मैरो चेंज करते हैं. Where you got this?
मैम मतलब कोई भी जेनेटिक डिजीज ब्लड के रिलेटेड होती है तो उसमें ऐसा होता है ना कि बोन मैरो चेंज करते हैं, तो मतलब वो
Do you
डिजीज
think
फ्रॉम
it's
रूट
possible? How will you change the bone marrow, beta? It's not
मैम
change.
मतलब बोन मैरो
It
ट्रांसफर
is,
करता
it
है?
is called bone marrow transplantation. It's a very slow process. Until date, we cannot say it is 100% efficient. There is a age in which it is partially efficient, but there is efficient and there is no guarantee for it.
तो मैम ऐसा WBC में नहीं हो सकता? जैसे अगर कोई एज से इफेक्टेड है तो जो WBC प्रोड्यूसिंग फैक्टर है अगर उसको ही ट्रांसप्लांट कर दे तो फिर
How many
risk क्या है?
WBCs do you think we have? You will run behind each and every WBC and replace them?
मैम WBC प्रोड्यूसिंग फैक्टर,
So what is the WBC producing sector? Where
मैम आई
WBC
डोंट नो
produce?
द नेम बट आई एम जस्ट सेइंग.
It is bone marrow only, beta. From the beginning I am telling you, all blood cells are produced in the bone marrow from the hematopoietic stem cell. Am I not speaking about this? I think I taught this to you when I taught you tissue. I taught this to you when I taught you circulatory system. Today also I started with this point that it is occurring from hematopoietic stem cell present in the bone marrow. Then what you are expecting? WBC will be produced from somewhere else, beta? Is it possible? No, na? Understood now why bone marrow is transplanted? Ayushi?
यस मैम. हाँ
That is the reason why bone marrow is transplanted, no? It is produced inside bone marrow only. So these WBCs can be further subdivided into two types. One is agranulocyte and the other one is granulocyte. So agranulocyte means they don't have granules present in their cytoplasm and they can be subdivided as lymphocyte and monocyte. Our granulocytes are the one which is having granule, and these granules are stained by different types of granular substances, and these, depending on the nature of these granule staining substances, we classify them as eosinophil, neutrophil, basophil. So if I am talking, eosinophil is stained by acidic substances, neutrophil by neutral stain, and basophil by basic stain. Okay, so leukocytes are 6,000 to 8,000 in number, and out of that, most amount is made up of granulocyte. So granulocytes are approximately making 72%. So I'm just writing it over here. So 6,000 to 8,000. Out of that, 78% is made up of granulocytes. And in granulocyte, the number, or rather to say the amount of this eosinophil is 2 to 3%. Okay, neutrophil is maximum, it is 60 to 65%. And if I'm talking about basophil, it is the least, that is 0.5 to 1%. Understood? So this is
Yes,
the
ma'am.
granulocyte. Apart from that, agranulocyte is approximately 28% of it, and out of that, the monocytes are like having 6 to 8%, and the lymphocytes are composed of 20 to 25%. Okay, so we will talk about each of them. Monocytes are largest cell, or out of all leukocytes, they are the largest cell, and they have a sub-rounded nucleus. See, if you look into the WBCs, they have various shape of nucleus, and that is the main important characteristics by which we can identify them. Last time when we saw the I taught you tissue chapter, at that time I showed you the blood smear diagram. I'm not showing them over here. So they are very much motile and they are phagocytic. So anywhere any cell debris is there, or antigen antibody complex is there, or dead cells are there, they simply go there and eat them up to clean the body. And that's why we call them macrophages. So generally, when they enter into tissue, these monocytes are converted into macrophages, and they, as the name itself is saying, macro means big and phages means which eats up everything. Next is about lymphocytes. They are having size of RBC, and they have a very large nucleus and small amount of cytoplasm. In your grade 12 syllabus, you will be having one chapter, which is chapter number 8. There you will study about function of lymphocytes, that it is the antibody producing cell or beta cell or B cell, or it can be acting as a T cell, that is the cells which are responsible to kill the altered cell like virus cell, or what I can say, the cancerous cell, they can. So basically they are very much important for killing the virally infected cell, as well as the different types of organisms enter into our body. Now I am talking about granulocytes, and first I wish to talk about the eosinophil. They are stained by acidic dyes, which is called eosin. Mama, so the eosinophil is stained by acidic dye called eosin, and they are having bilobed nucleus. That means if you see their nucleus in the cell, if this is the cell, the nucleus will be having this kind of lobes. So by looking into the structure of nucleus, you can easily identify the eosinophil, and they are full of acidic granules which can be stained by acidic stain, and these granules are full of hydrolytic enzymes, and they are having peroxidase, which may be responsible to be released in the phagosome. So what is the function of eosinophil? Whenever we are having allergy in our body, any kind of allergy due to presence of any living organism, or it may be any non-living organism like dust, dirt, particle, or anything, we have certain reaction in our body. For example, there may be redness, itching, swelling, and all these reactions are due to presence of histamine. So we say that allergy is good because it is drawing attention of immune system as well as drawing attention of ours to that area. But we cannot tolerate that pain forever. No, something should be there in our body which should reduce the pain. So there comes the role of eosinophil. Whenever there is any kind of allergy, the ESR count is very much required. That's why it is said that TCDC, ESR. These are the three common blood cells, like blood test which is done by generally practitioner. That is the total count, direct count, and the eosinophil sedimentation rate. So if a person is having any kind of invasion or allergy, the eosinophil number increase. Why? They have property which is called antihistamine property, and they help us to fight against the disorder. So if you look into the blood of a person who is asthmatic or having hay fever, you will be seeing always more eosinophil count in them. Apart from that, eosinophil have a very unique function. It can be very helpful in dissolving the blood clot. We always talk about blood clot, blood clot. You know how dangerous they can be because they can block the pathway of the flow of the blood. So if it is formed in small quantity, eosinophil can dissolve it. Now I wish to talk about neutrophil. As their name suggests, they can be stained by acidic stain as well as basic stain. So that's why we call them neutrophil. And they have many lobed nucleus. If you see the structure of neutrophil, they have minimum 4 lobes or segments in the nucleus. So that is how you can identify the structure of the neutrophil. You can identify, okay, this is neutrophil. They are having azurophilic granules, and later on this azurophilic granules will be converted into hydrolytic enzyme. So what is their role? Their role is mainly phagocyte. Neutrophil like monocytes help in engulfing the microbes, and they actually attract the bacterial cells. Once in our body the bacterial cells are there, they will go there and they will kill them by secreting the bacterial peptides. Last but not least, the basophil. So what is their function? They are stained by basic stain which is called methylene blue, and they are very large. They have S-shaped nucleus. So granules are full of histamine, and basophil are responsible to produce histamine and heparin. So histamine is responsible for giving us the allergic response, and heparin, you know that it is the anticoagulant present in our body. Okay, so generally platelets, after this we will talk about the platelets. So platelet is the third type of blood cell that is present in our body. So it has also a good name. Can anyone tell me the good name of platelet?
Thrombocytes.
Thrombocytes. So they are generally rounded or oval disc-shaped type structure, and they are very small, in fact smallest element of the blood. So their number should be like I should write it. It is approximately 250. This is the platelet per cubic millimeter of the blood, which is quite high. And similar way, the way leukocytosis, erythrocytosis is there, their number, if it is increasing, we call it thrombocytosis, and if it is decreasing, we call it thrombocytopenia. And they are colorless, they are flat structure. Apart from RBC, platelets are also non-nucleated, and they are equally produced at the bone marrow itself. They are also produced by the bone marrow. So whenever we are injured and our blood needs to be clot, at that time platelets come in the picture, and they release platelet factors or thromboplastin, which help the blood to clot. They have 3 to 7 days of life period, and like WBC, they are also phagocytosis by the phagocytosized, rather to say, by in the blood cells. One, their lifespan is done. Okay. So I just wish to add a very important thing over here. The hematopoiesis. I told you that pluripotent stem cell is responsible for the process of hematopoiesis. So bone marrow is having all the factors and microenvironment where where the whole process of cell production is possible. And the stem cell first form the lymphoid stem cell and myeloid stem cell. From lymphoid stem cell, the lymphocytes are produced, and from myeloid stem cell, all other types of blood cells are produced. Understood? Is it clear? Class? Are you there? So that is all about blood. If I wish to ask you a question, do you have any doubts regarding blood grouping? I think we discussed about it very well in detail about AB blood grouping. I taught you about the gene and their transmission also. We performed a few experimental case study also with your own families. So is there any doubt in blood grouping?
No.
No, ma'am.
Great. So just want to highlight, I guess all of you know what is erythroblastosis fetalis.
Yes.
So now quickly I wish to talk about the blood clotting. So do you have any doubt regarding that blood clotting or clotting of blood?
No, ma'am.
No one? So whenever we have injury in our body, just wish to mention, there is a vasoconstriction. Blood clotting, so we know that it start with the, whenever there is any kind of injury, it start with the thromboplastin, which then go for cascade of reaction with different steps to finally form the fibrin, which is a net-like substance which will block the blood to come out of the blood vessel, and we are protected. So the thing is that there is another role of serotonin which I just want to say. Serotonin is a very good vasoconstrictor. The moment there is a blood vessel is broken, the place where the blood vessel is broken, it goes there and promote vasoconstriction only to that place. Why only to that place? Because agar all over vasoconstriction hua, then blood pressure will increase and blood flow will be more, blood loss will be more. So it will go there into that specific place and make the vasoconstriction, which will be very helpful for us to get saved. Okay, so this is something extra I wish to say. what do we mean by thrombosis? Have you ever heard about this coronary thrombosis or any kind of thrombosis? See, we have heparin, which is the natural anticoagulant present in our body, and it never allows to blood gets coagulated in our body. But then also we talk so many times that thrombosis is there, blood clot is formed, and this type of clots are called thrombus, and the process of formation of them is called thrombosis. So why do you think there can be formation of this type of blood clot? Sometimes blood vessels are having internal injury which may cause the formation of blood clots, and these are very dangerous. In grade 12, we will know that some microbes can help us to fight against these clots, and we call them clot busters. Okay, so if you remember, while I was teaching you classification of animal kingdom, other than this heparin present in our body, we talked about a specific type of blood coagulant present in animal system secreted by a special animal. Can you mention that?
Hirudinaria.
Hirudinarin, secreted by Hirudinaria granulosa. Okay. So I wish to conclude today's lecture with difference. I wish to talk about the difference of blood plasma and serum. I told you at the initial stage of this lecture. See, serum and blood plasma, are they same or not? Blood plasma is what we studied already, the composition, everything we studied. What is serum? Whenever we have clotting of blood, colorless liquid is released from that place. We call it as blood serum. So is plasma and serum same? No. Blood plasma is yellowish fluid, whereas serum is a clear fluid. It has red blood, all types of corpuscles, that is the WBC, RBC, platelets, and fibrinogen. In case of serum, it neither has the blood cells or has the fibrinogen. Blood plasma, as has fibrinogen and all cells, it can clot, but blood serum can never ever clot. So there are actually 13 clotting factors present in the blood, starting from fibrinogen, prothrombin, thromboplastin. Calcium ion plays a very important role in conversion of prothrombin to thrombin. So it is a very important thing. There can be proaxillerin, which is a globular protein, which we call as labile factor. Then there is sixth factor, which is hypothetical. It's not isolated. There is PICA, that is serum prothrombin conversion accelerator. So as you can understand, these accelerators are responsible to accelerate the clotting condition, and they are obviously under control. Apart from that, the eighth factor, that is AHF, that is anti-hemophilic factor. This factor is missing in case of person who are suffering from hemophilia. So once again, I'll repeat what happens in hemophilia. In hemophilia, the clotting ability of the person will be gone. So what will happen? Even if they have smallest of the cut, they are not able to clot the blood. So blood will flow and you have to immediately hospitalized, otherwise there will be problem. Next is the ninth factor, which is also known as Christmas factor. The good name of this factor is plasma thromboplastin component, and this thromboplastin component is responsible for making thromboplastin. Then there is Stewart factor, there is PTA, PTC is plasma thromboplastin component, and the PTA full form is plasma thromboplastin annexidin. So both of them are working antagonistically. Then there is Hegman factor, and last the 13th factor is called fibrin stabilization factor or Lackey-Leonard factor. This 13th factor is responsible for stabilization of the the fibrin produced, which is finally going to stop the flow of blood. Apart from that, if I go back to the blood vascular system, there will be heart, which is 4-chambered in us, and that's why we have proper double circulation and blood is not mixed. This is not the case in case of fish and amphibian. They have mixed blood. Although they have closed circulatory system, they have mixed kind of blood. And only out of amphibian and then reptiles also have mixed blood. Only birds and this one, the mammals are having oxygenated, deoxygenated bloods transported separately. But while you are mentioning about reptile, you should always mention that the crocodile is having the 4-chambered heart. Okay, so human heart is that the one. Then the blood vessels can be further subdivided into the vein, artery, and blood capillary. These parts are already taught to you in detail, so kindly go through them, and I hope you will be able to understand. So today that's all from my side. Bye.